Okay, so good afternoon, everyone. And first of all, I would like to thank Carol, Frank, and Xtraordinaire for organizing this amazing symposium, and all of the families for coming here from different parts of the world. So today with my talk, I'm going to try to convince you that functional near-infrared spectroscopy can be one neuroimaging tool to develop a functional biomarker for creatine deficiency syndromes. So let me give you a little bit of context. As many of the speakers basically already mentioned also during this morning, there are many preclinical trials ongoing for GAM deficiency and CTD, and clinical trials are on the horizon. But one additional problem that we have to face in CTD or CTDS research is the scarcity of unbiased and quantitative functional biomarkers for monitoring the activity of brain cells. Slowly, okay, for sure. I'm strongly convinced that if you are going to sit on the same bunch of behavioral endpoints to assess drug efficacy in preclinical trials, we are going to face the risk of misinterpreting our results, basically because the phenotypic observation of behavioral endpoints can be really prone to subjective bias. And I guess that at least, well, wet scientists can agree with me that there is a need of innovation in trial design and analysis, and that the success of drug testing strongly relies on the availability of objective readout, so to bring out the potential benefits of transformative therapies. And the same is true also for clinical trials, because the combination of behavioral and functional measures can get us a more comprehensive reading of what the drug is doing on the brain cells and on patients, of course. So this is why one of the main goals in the lab in the last few years has been to identify at least one of these biomarkers. Of course, we cannot use just one. We need a panel of biomarkers, and we have seen beautiful presentations before of possible other measurements. But we would like to identify at least one biomarker, starting from the study of the preclinical mouse model of CTD that we have previously generated and extensively characterized at the behavioral and biochemical level, but also working with patients. So nowadays, there are multiple techniques to visualize the activity of neural cells in the living brain, including, for example, as already mentioned, functional MRI, PET scanning, ultrasounds, and EEG. But in the last few years, a very creative postdoc in the lab, Raffaele Mazziotti, developed a custom system of intrinsic optical imaging, which is a non-invasive technique allowing to measure hemodynamic responses in the brain. IOS is non-invasive because it relies on the capability of the red light to penetrate the skull and basically see what happens in the brain without making any major surgery in the animals. This IOS signal is very similar to the bold signal that you get with functional MRI, because the two main sources of the IOS are the transient changes of hemoglobin oxygenation in the active brain tissues and the subsequent changes in the local blood flow. As you can see from the GIF, you see that active brain areas appear darker than the surrounding regions, and this is because they basically reflect less of the red light that we are going to use for eliciting a response. And this decreased reflectance basically produces the negative signal that you can see on the left of the GIF. And so please keep in mind that IOS produces negative signals, because this is important for the other slides. Okay, so since metabolic alterations produced by creatine depletion can strongly affect the hemodynamic responses in the brain, we decided to use this technique in our CTD mouse model. And so Giulia Sagona, at the time a PhD student in the lab, she is now a very talented postdoc, did this experiment. So basically, she performed IOS imaging across the disease progression in the CTD mouse model at 3 different times, P40, P110, and P180. And what she found is that there is a consistent increase in the amplitude of IOS signals in the brain of knockout mice with respect to wild-type animals. We did not record resting state signals, but we recorded evoked responses, and we did with visually evoked responses, because there is growing evidence in the literature pointing to a visual impairment across multiple neurodevelopmental disorders and to visually evoked responses as a good proxy for assessing the activity of cerebral cortex and more in general of the whole brain. So as you can see from the left graph, there is an increase in the absolute amplitude in the brain of knockout animals, indicated by the blue dots with respect to the gray dots, which are the wild type. Remember that the signal are negative, so lower dots means higher amplitude. So these results basically demonstrated that the amplitude of the signal can effectively discriminate between wild type and knockouts. And here I reported also the data about peak latency, showing that also the peak latency is increased in knockout mice, at least P110 and P180. So basically, altogether, these results demonstrate that iOS can discriminate between the two conditions, wild type and knockouts, and support the idea that cerebral oxygen consumption can be a very useful biomarker for disease prediction. Actually, we have done the experiments in males so far. We are now doing females, but I don't have any results to show, hopefully in the next few months. Julia performed, at P40 and P110, Julia performed the iOS imaging along with the assessment of psychomotor function in the mice using 3 different behavioral tests that are commonly employed to characterize the endopathological phenotype of these mice, which are the grip strength, which is basically a measure of muscle function, the YMAs, the performance in which is strongly related with cognitive function, in particular with implicit working memory, and the rotarod, which is a task commonly used to assess motor coordination, but also to investigate the presence of autistic-like stereotype movements. And what she found is that there is a significant correlation, at least for the rotarod and the YMAs, between the magnitude of iOS responses and the behavioral performance of the animals, with the higher being the amplitude of iOS and the poorer being the behavioral performance of the animals, so suggesting that the level of hemodynamic alteration could be predictive of the severity of symptoms. The correlation is somehow lost at P110, but we think that this is due to the longitudinal nature of our protocol, because you can see that, at least again for the YMAs and the rotarod, the linear regression are not so bad. So this is why we are doing now a separate group of animals just dedicated to this time point to see whether we can replicate the same results seen at P40. And this is still work in progress also because we would like to expand the number of behavioral testing that we are going to correlate to the iOS imaging to see whether we could find a correlation with other functional domains, such as, for example, declarative memories. Then Julia also asked whether the iOS biomarker could be sensitive to a treatment. And she used cyclocreatine that Dr. Longo mentioned this morning, which is a lipophilic analog of creatine, the development of which was discontinued because of the toxicity that he mentioned. But this oral treatment with cyclocreatine has shown some partial positive results on the cognitive and autistic-like phenotype in CTD mice. And so we decided to test whether there was an effect also in iOS imaging, and the answer is yes, because, well, there are no asterisks here. I don't know why, but there is a significant attenuation of visually evoked iOS imaging in the animals treated with cyclocreatine with respect to untreated mice and vehicle-treated animals. So basically the answer is yes, the biomarker is sensitive to the treatment. And more recently, we also had the opportunity to perform some iOS imaging in a Gantt mouse model, thanks to the collaboration with the group of Mauro Magnani at the University of Urbino. And, well, the sample size, you can see, is quite small so far, but we have these preliminary results that seem quite encouraging, showing an increased amplitude of iOS even in the GAMP mouse model. This is not so surprising to me because I think that the hemodynamic alteration is due to the lack of creatine in the brain, and of course also in GAMP there is creatine depletion. We have, of course, to increase our numbers, but well, it seems that the validity of the biomarker can be easily expanded also to GAMP deficiency. So let me just very briefly summarize what we have found in the mice. Basically, it seems that iOS imaging has the 3 ideal features for a biomarker. Can be disease predictive, so can classify wild type and knockouts, can be predictive of the severity of symptoms because there is a correlation between the numbers emerging from iOS analysis and the numbers emerging from behavioral performance, and it's sensitive to the treatment, so it is sensitive to assess the efficiency of treatment-induced recovery in neural circuit function. So far so good, everything is fine, but these are only mice, and so the main question is, what is the translational value of this method? Well, the answer is it's pretty high because there is this technique that I already mentioned during my first slide, which is called functional near-infrared spectroscopy, or FNIRS for friends, allowing to perform the very same measurements that we have done in mice with iOS in humans. And well, you can see here that NIRS is a totally non-invasive technique, just a cap-based bunch of optodes placed on the head that can be easily used not only in adults but also in younger people. So thanks to the collaboration with the Stella Maris Institute, which is a neuropediatric hospital close to Pisa, where I had the opportunity to work with patients, we acquired one of these systems and we started developing the setup with the recordings of adult volunteers and children with neurotypical development. And Elena Scaffei, which is a very talented child neurologist, which is doing the PhD in my lab, basically, using FNIRS, started this process, and she immediately realized that the classic paradigm for visual stimulation, consisting in the combination between a white and the black checkerboard and the gray baseline, which is, of course, can be easily used in anesthetized mice, can be quite boring for awake people. Because, this is also because the hemodynamic responses are quite slow and the size of the signal is quite small, so it requires the averaging of multiple trials consisting of 5 seconds of the checkerboard and 10 seconds of the gray baseline. So the whole protocol lasts 10 minutes, which is not so much per se, but well, staring at the gray screen for 10 minutes without doing anything else can be very unpleasant. So we decided to try to increase the entertaining value of our stimulus, and we blended the classical visual stimulus with the cartoon, with the commercial cartoon. And here, I'm trying to show you how the stimulus looks like. So you can see that there is the cartoon in grayscale with the audio on, and even when the checkerboard appears, the blending procedure allows us to still resolve the contour of the profile of the characters, so you can still follow the storyline quite well. And so we used, let me stop the cartoon, sorry. So we used this kind of stimuli, and we decided to compare in adults and children the amplitude of responses following the classic visual stimulus and this cartoon-based stimulus. And these are, these are the results that we got in adults. In the panel A, basically you have the response following the classic visual stimulation, and as expected, there is a positive deflection, so an increase in total hemoglobin and oxygenated hemoglobin, and a decrease, a lower modulation of the oxygenated hemoglobin, which is, well, quite well, it was expected because more activity requires more oxygen, okay? But the same is true for the other two conditions described in panel B and panel C, where we use the cartoon-based stimuli. So we have seen the very same modulation of total and oxygenated and the oxygenated hemoglobin. And the other important thing is that the amplitude of the response was not affected by the presence of the cartoon, but also by the cartoon that you choose to be displayed with the stimulus. Indeed, well, I'm sorry I don't have a pointer, but in the panel D, you can see that basically the amplitude of the responses among the 3 different conditions are quite comparable, except for a small decrease in the amplitude of the oxygenated hemoglobin, which is in any case the less informative metrics according to the literature. And we also checked whether the contrast level at which we decided to show the cartoon was another potential confounding effect for the analysis of the amplitude of response, and the answer is no, because we tried in a separate group of subjects to display the cartoon at the 20, 40, and 80% of contrast, and again, the amplitude of the signal was still comparable. And then these are the results about the children. Basically, they tell us the same information. We didn't ask to the children to see the boring visual stimulus, but they just watched 2 different cartoons at 2 different levels of contrast, but the basic take-home message is the same. So the amplitude, there is a reliable response to the stimulus. The amplitude is the same. The amplitude is not affected by the cartoon chosen and not affected by the contrast level. So basically, we have a novel visual stimulus with high entertaining value, and you basically can choose any kind of video, movie, or cartoon to be blended with the checkerboard. The only important information is in the panel A of the right figure, is that the amplitude of FNIRS responses is higher in children than in adults, and this is probably because of the progressive thickening of the skull during the age. But this is an important information to keep in mind for the next slide. So these results prompted us to start at least a pilot study in CTD patients so far, with a twofold aim. The first one was to test the feasibility of visual FNIRS recording in this clinical population, and the second was to see whether the amplitude of FNIRS recordings could discriminate between patients and typical controls. So this is the table listing the basic information about the 11 CTD patients diagnosed at the Stella Maris Institute. Unfortunately, we lost 4 of them due to family and logistic reasons at the recruitment phase. The answer of 2 of them is still pending, and we recorded so far just 5 CTD patients, and these are the 5 CTD heroes that already came to Pisa for doing the recordings. But as you can see from the age column, there is a strong heterogeneity in the age at which the recording was done. And this might introduce, if you remember what I said a few seconds ago, might introduce a very important confounding effect in the analysis of the amplitude. And so we decided to analyze them so with a further division into groups according to age. So we divided this very small sample size in 2 subgroups, the first one of people over 18 years of age, and the second one of children below 18 years of age. But as you can see, in this, doing this, our sample size is basically nothing. But in any case, we have some preliminary results that I want to show you. Here you have the results in children, showing that basically we have a reliable response to the checkerboard with respect to the baseline. You have in green for controls and in red for CTD the response to the checkerboard, and in gray the response to the baseline. So you can see that there is an increase in total and oxygenated hemoglobin, which is significant. Well, not significant because we have just 3 controls versus 2 CTD, but definitely higher than the baseline. These are the same results in adults, in the adult subgroup, which are less clear because of the poor quality of one recording. But the most important thing is that comparing the response to the checkerboard between CTD and controls, you can clearly see that in the young population, there is a strong tendency towards an increased amplitude of the responses in CTD individuals with respect to controls. And the same is true also for adults, despite the confounding effects of some not so clear recording. So these are of course, just preliminary results, very far from being conclusive, but let me just show you a few take-home messages. The first one is that we devised this innovative protocol for visual stimulation with high entertaining value, which can be used for FNIRS recording, but of course can be extended to other, I don't know, EEG recording or fFMRI setups. We tested and demonstrated, in my opinion, the feasibility of FNIRS recording in the CTD population. I guess that will be the same for GAMT. And we have preliminary results suggesting that CTD patients show a higher amplitude of dynamic responses with respect to age-matched controls. But of course, we have a very small sample size. We need to go multi-site to demonstrate something. And we have been lucky because this project has been recently funded by an European grant. So we will have the opportunity to perform parallel recordings at the Hospice Civil de Lyon, thanks to the collaboration of Dr. Aurore Curie. And this is a very fresh news. The same project has been also funded by a very small grant, a very small internal grant of Boston Children's Hospital, Harvard Medical School. So we'll bring the study also in the US, the Boston Children's Hospital, thanks to the collaboration with Dr. Michela Fagiolini and Dr. Irina Alsen, but also at the NIH with Danny Porter and Samar Rahal. And so I hope to have more results in the next year or so. So this is basically the end of the story, and this is the list of people working with me in creating a related project. Here I would like in particular to mention and thank for their effort Elena, Giulia, and Raffaele, because they worked on the biomarker project, but of course a special thanks goes to all the people working with me. This is the list of collaborators and funding agency allowing us to do this research, and of course a special thanks goes to Xtraordinaire and ACD and also the Italian families because well, they constantly support our research. And thank you for your attention.